Hello students, welcome to today's class. Today's class will be dealing with the one more topic in our BSc Degree Botany Semester 3 paper code BSc BOC 231, paper name Pteridophytes, Gymnosperms and Angiosperm Embryology, under that Embryology of Angiosperms. In this particular unit we will be dealing with the topic development of male reproductive structures. As this picture can show you that there are reproductive structures, and these reproductive structures are called as, this is female reproductive structure, there is a stigma and this is style, and whereas these are the anthers and filaments. And how the male reproductive structure, that is pollen grain, will develop, that is what we are going to see in today's class. In a angiosperm, characteristic feature of them is the production of flower. Flower is said to be modified shoot for the purpose of sexual reproduction. Here we can come across two types of whorls, namely accessory whorls and essential whorls. Among them, the calyx and corolla, which is outermost whorl, calyx, which is made up of sepals, and corolla, inner to that, made up of petals, are called as accessory whorls, whereas androecium, which is represented by stamens, individual units of the androecium are called as stamens, and gynoecium, individual units are called as pistils, are termed as essential whorls because they directly involve in the process of sexual reproduction, for which the flower is meant for. Among these two essential whorls, the male reproductive system, androecium, that is a third whorl from the outside and second whorl from the inside, is made up of individual segments which are called as stamens, and one stamen is represented by characteristic three components, namely a long stalk-like structure which is called as filament, and anthers, which are flattened or bulged structure within which we can see the pollen grains growing. And there is a region where the anthers are connected to pollen, and that particular connected to filament, and that particular region is called as connective. So this is with respect to the male reproductive structure and structure of anther. And to furnish that particular detail in still more detailed manner, here we have taken a photograph from the electron micrograph or high definition micrograph of the anther lobe, where you can observe in a very clear manner there is a region where the filament is attached to the anther lobes. These are all the anthers, and this is a stalk-like structure. These are all anther lobes, and this is a stalk-like structure which is called as filament. And when it comes to these globular structures, spherical structures, these are called as pollen grains, which will be produced within these pollen chambers. And pollen chambers are called as microsporangia. Within that, microspores will be developed. And these are the external structure, anther lobes, and these are variedly colored, and their fixation to the filaments will be different in different families and plants. So this is a detailed version about the stamen, structure of stamen. So there will be filament, and there will be anther lobes which are connected by a region called connective, and within the anther lobes pollen chambers we can come across pollen grains, and the anther lobes are elongated large inflattened structures comprising of these microsporangia. Usually anther lobes will be two in number, and individual anther lobes will consist of two chambers, but it can vary from one to two like that. When we study the structure of young anther in its transverse view, we can observe the detailed structural components in a nice manner, and this particular diagram shows those things in a very proper manner. And if you can observe the external appearance of this particular section, we can see there are clear cut two lobes within this particular diagram and or section, and individual lobe, and these two lobes, each lobe will represent one anther. So since there are two anther lobes and one anther lobe is represented in this particular section. And this particular each lobe represents two chambers within that, and this is called as bythecous tetralobed condition in a technical manner. But if you can remember, even if you can remember that there are two chambers or two lobes for that matter with two chambers in each lobe, so totally we can see four chambers in this particular section. What are these chambers? Why they are there? So actually these are the pollen chambers where exactly the male reproductive or microspores are going to get produced in the plant. That means this is the site, these are the sites where actual meiosis takes place in the male reproductive structure of the flower. And this particular structures will be accompanied by certain nutritious structures like tapetum and certain protective structures, and particularly there are structures which will be helpful for their dehiscence or their dispersal or to facilitate their pollination in once they get matured. And to understand these particular things, let us study different layers one by one. So keenly observe, if you keenly observe the external outermost layer, there is a single layered cells, uniseriate cells are there which are surrounding whole of this particular structure, that is the anther, and that particular structure is called as epidermis. Epidermis is a uniseriate or single celled thickness having structure. And next to that, what we can observe? We can observe there are certain cells. If you observe individual cell, if you observe these cells, they are not normal cells. That means they have been situated little bit elongated in their nature, right? Can you observe these things? So these elongated structural components or cellular structures which are situated next to epidermis are called as endothecium. Next to that, if you observe throughout the body, those structures are there except in these regions, right? So you can observe the endothelial cells are present within this particular structure. And next to that, if you observe, there are again parallelly laid cells, 2 to 3 layers, 1 2 3 3 or 2 to 3 layers of cells. Sometimes there will be more, sometimes there will be less, but you can observe very keenly. If you can observe very keenly, you can they are very much appear in the manner of parallelly laid cells, and this particular layer is called as middle layer. What it is called as? This particular cells are called as middle layer cells. And all these three layers, basically all these three layers are protective in nature, and these two layers are originated by the outer cell of the sporogenous tissue. That means in the beginning there will be only a small mass of cell in the torus or thalamus region, and later that mass of region will start appearing, protruding out by the production of the filaments. Later, at the terminal region of the filaments, anther lobes will be formed, and those anther lobes at the beginning they will contain sporogenous tissues within them. And sporogenous tissue are derived from the archisporial cells, and archisporial cell outer cell will give rise to middle layer and endothelium, whereas inner cell give rise to two very important components, namely tapetum and pollen mother cell. What is this tapetum? You can observe this. The dispersion or the distribution of the tapetum is, as the maturity of these inner cells are more, you can see less amount of tapetum or consumed tapetum. So here basically tapetum act as a nourishing tissue. That means for the cells which are undergoing in the transformation stage to form the microspores, from their microspore mother cells, these cells will be giving all type of nourishment for that developing tissues. And what are pollen mother cells? Pollen mother cells are the cells, you can see different kind of divisions are taking place. All whatever the stained portion within those cells, it is depicting the different phases of the cell division. And this here, the pollen mother cell will undergo meiotic division to form the pollen tetrads. All these processes will take place inside this. So the conversion or transformation of the sporophytic generation, that is 2n condition, in which spore mother cell or pollen mother cell will be there, it will get converted into gametophytic generation within these pollen chambers. And these chambers are also occupied, connected with the connective, which connects to lobes. And very importantly, we have to study that stromium. Stromium is a region which will be facilitating the dispersal. Once the microspores or pollens are formed within the young anther, what happens? These things will dry off, and this particular region, since there is a less tissue differentiation, it will split off, and pollens will be easily available for its pollinators. So this is with respect to the structure of young anther. So you have to remember all these different types of parts in their respective areas. So when the pollen grain gets matured, there will be pollen tetrads will be formed into pollen mother cell will transform into pollen tetrads, and all other forms will start disappearing one by one, or mature stamen mature anther lobes will show much more differentiated, much more profound structures than the young anthers. Otherwise all the structures almost they will be similar, except in the young anther we can observe pollen mother cell, whereas in the mature anther we can observe pollen tetrads. Hope this particular lecture was useful to you. Thank you for listening.